Chapter 5 focuses on plasma membranes and how things move in and out of the cell. We talked about the fact that cells are small. Most of them have to be seen with a light microscope. And the reason why cells are small is in order to move in enough materials that the cells need, the good stuff like oxygen and glucose, and move out the waste products, things like carbon dioxide and ammonia. And once a cell gets too large, it can no longer efficiently move in enough good stuff and move out the waste products. So this chapter will help us understand how things move around cells. Okay, this chapter focuses on the blank membrane, which is the boundary that separates a living cell from the surroundings. This blank membrane exhibits selective permeability, allowing some substances to cross it more easily than others. And this blank membrane has proteins that span it to allow some substances to cross it, but not others. Okay. This book, and I will try to use the word plasma membrane, P-L-A-S-M-A. Sometimes you'll see the outer membrane called the cell membrane, but I think plasma membrane is more specific. There are a lot of different membranes within eukaryotic cells. All the organelles have membranes around them. So when we talk about the plasma membrane, I think it's easier to see that's specific to the outer membrane that separates the cell from its external environment. Here are some of the functions of the plasma membrane. It defines the border of the cell. It manages what enters and exits the cell. It receives signals from outside and initiates cellular responses, and it joins together neighboring cells to form tissues. Here are some questions to see what your background is and get you oriented towards the kind of things we'll cover in this chapter. The molecule shown at right, this molecule right there, or in a phospholipid bilayer, it's right here. What does that molecule do? Okay, and both A and B are true. It's a phospholipid. The top part is charged. The bottom part is a lipid that's nonpolar or uncharged. And so when it forms a bilayer, the part that's charged is on the outside attracted to the water, and the parts that are uncharged and repel the water are on the inside of the bilayer facing one another. Okay, question 2. What's the best caption for the figure below? Is it that eukaryotes have membrane-enclosed organelles, but prokaryotes do not? Is it that eukaryotic cells are 10 times larger than prokaryotes? Or is it that the plasma membrane is the barrier that separates the inside and outside of the cell? Okay, so A, B, and C are all true, but this figure is illustrating C. It's showing the plasma membrane, which is the barrier that separates the inside and outside of the cell. Okay, question 3. Look at the figure and tell me what the best caption is. Is it that proteins for export are made in the ER membrane and secreted by exocytosis? Is it that plasma membrane is manufactured in the ER membrane and moves to the surface during exocytosis? Or both A and B are good captions? And the answer is C. So ER membrane makes proteins to be exported, like number B, like number A, but it also makes plasma membrane, like number B. So both A and B are true. So C is the correct answer. Here's a video you'll want to look at by Ricochet Science on plasma membranes. Okay, the model of how the plasma membrane works is called the fluid mosaic. The red part of the model represents a phospholipid bilayer, and the phospholipid bilayer has a phosphate head, which is charged and attracted to the water. the fatty acid tails are nonpolar and orient themselves away from the water. And so it's like an oil droplet that surrounds every cell. And that oil droplet is a barrier to things moving in and out of the cell. If that oil droplet is a barrier, then how do things cross over the cell? There are proteins that are membrane-spanning proteins. Sometimes they're called integral proteins if they cross the phospholipid bilayer. And some act as channels to move things in and out across the cell. They have other functions as well. Some are receptors, and there'll be a receptor to a molecule that hits the outside of the cell, like ringing a doorbell. And there'll be a response at the inside of the cell. And some are glycoproteins and glycolipids, which means sometimes there are glucose groups or carbohydrates attached. And when there are carbohydrates attached, either attached to a protein or attached to the lipid part of the membrane, the glucose molecules or the carbohydrates are recognition molecules. Here's a blow-up of how a phospholipid molecule works. A phospholipid molecule is amphiphilic. Amphi means both. Philic is loving, like the city of Philadelphia is a city of brotherly love. So the head is charged, the tails are uncharged, and so that's why they form the phospholipid bilayer, where the heads point out towards the water and the tails point in towards one another, the uncharged part. Okay, so after the lipids, the second major component of membranes are these proteins. And if they go all the way across, they're called an integral protein. Sometimes they're peripheral proteins. So a peripheral protein might be a place for the cytoskeleton to bind to. It might be structural. There are other functions of peripheral proteins as well. Some of them will act as enzymes. About a third of all the proteins in a cell are bound to membranes. So there are a lot of proteins that float freely in the cytoplasm, but there are a lot of proteins that are bound to membranes as well. This is showing some different types of integral proteins. The integral proteins will extend all the way across the phospholipid bilayer, and they're just telling you that integral proteins will often orient themselves the same way the phospholipids do, with the charged molecules on the outside and the hydrophobic or uncharged parts on the inside. And they're telling you, well, how can proteins be charged and uncharged? It depends on the amino acid side chains. And so just dividing them up by the side chain charges. So some side chains are uncharged, nonpolar. Some are partially charged, so these are called polar. And some have complete charges. So here are negatively charged ones, and here are positively charged ones, and here are some more nonpolar ones. All right, so the third membrane component are carbohydrates. Carbohydrates are good recognition molecules. Sometimes carbohydrates bind to proteins and form glycoproteins. Glyco is a word for glucose. Sometimes carbohydrates bind to the lipid directly and form glycolipids. Later on, we'll come back to blood types, A, B, and O blood type, and A, B, and O blood type differ because of the carbohydrate that's bound to those red blood cells and how the immune system recognizes them as self or non-self. Carbohydrates can bind together in different ways to form these different patterns, and that's why they're easy to recognize. Kind of like when you drop a letter in the mailbox. If you write an address on it, it will get somewhere. If you don't write an address on it, nobody will know where it's going. So the address is written by the carbohydrate pattern. This is a really interesting slide. It's showing HIV, the virus that causes AIDS. In order for HIV to get into cells, it binds to a glycoprotein. That glycoprotein is called CD4. And that glycoprotein recognizes HIV as being self instead of foreign. And so the HIV virus will enter a T cell and destroy that T cell, which is part of your immune system. Now, people who have European ancestry, about 10% of them will not get AIDS. If HIV enters their blood, they don't have the CD4 receptor, and so the AIDS can get into their plasma, but it can't get into the cells, into the T cells, because there are no recognition sites that say, hey, that's part of self. This may have been something selected for thousands of years ago by a different plague, some other disease, and it seems to confer resistance to AIDS. So sometimes we have traces of our ancestry within our DNA. Some of them are obvious, and some of them are things that we can't really see, but they're there under certain circumstances. Okay. The membrane should be as fluid as salad dressing. The way it regulates its fluidity is a few different ways. Plant cells tend to regulate their fluidity by phospholipid type. The more unsaturated the lipids are, the more fluid it will be. So plants that live in colder environments have more unsaturated lipids. They tend to be more fluid. Membranes that are in hotter climates, things like palm trees and coconut oil, tend to have more saturated fats, which are less fluid. Now, animal cells act differently. They have a buffer in their membranes called cholesterol. So we often think of cholesterol as being unhealthy, and apparently too much dietary cholesterol can be unhealthy, but cholesterol that we make and is put into our membranes is part of every animal cell, and it keeps the membrane more fluid when the temperatures get colder, and it tends to hold the membranes together, keeps them a little less fluid as the temperature gets warmer. This is a nice video of membrane fluidity, and so look at your downloaded PowerPoints and click on it, and it will show how the membrane is fluid. This is showing the fact that plasma membranes are asymmetric. The carbohydrates that are being recognized by the immune system are on the outside. Other proteins and carbohydrates would be on the inside. It's kind of like, would you ever put your doorbell on the inside of the house? No, it wouldn't make any sense. It's on the outside, so people will ring it, and then inside the house is the dinger that makes the noise and you respond to. It's the same with the plasma membrane. There are components that are on the outside, and there are components on the inside. We say that the plasma membrane is selectively permeable. So some things cross it and some things don't, and the proteins regulate what crosses and what doesn't cross. When things cross it, sometimes it's just natural passive forces that allow things to cross the membrane, and sometimes there's actually energy expended. So we'll talk about passive and active transport across the plasma membrane. In passive transport, there's no energy required, and diffusion is the simplest form of passive transport. When you talk about diffusion in membranes, we're talking about things moving right across the lipid bilayer. Diffusion is defined as the movement of substances from an area of higher concentration to an area of lower concentration. And we use this principle in our everyday lives. If you've ever put on cologne or perfume, it moves from you, where there's a lot of it, into the surrounding area where there's less of it. If you ever pop popcorn, you put the popcorn in one place in the room, but pretty soon you smell it everywhere. And so that's just a natural process of molecules bumping around, and they bump from places where there are a lot of them to where there are fewer of them. So if the holes in the plasma membrane are small enough for these particles to move across, and if they're uncharged particles and aren't going to get repelled by the plasma membrane, then they can sneak through the phospholipid bilayer, and they'll sneak through from where there are more of them to where there are less of them until they're at equilibrium. At equilibrium, they'll move back and forth equally, so there will be no net change. Small molecules that will do this are oxygen and carbon dioxide. So carbon dioxide and oxygen are moving in and out of cells just by simple diffusion across the plasma membrane. The biggest factor that's going to affect diffusion rate is the concentration gradient. It's molecules are going to move from where there are more of them to where there are less of them. Temperature will also affect it. Molecules move faster at higher temperatures. The more surface area to diffuse across, the greater the rate of diffusion. The greater the distance, the slower the rate of diffusion. And sometimes there's pressure involved, like in kidney cells, where blood pressure will force diffusion rates to increase. Okay, facilitated diffusion. Facilitated diffusion is also passive transport, and it's called facilitative diffusion because things are moving by diffusion, but they're moving through a protein. And the things that will diffuse this way are ions, so things that are charged like Na plus and K positive, Cl negative, and small polar molecules. So glucose is one we'll talk about, and it has a hydroxyl group. It has a few hydroxyl groups. OH share electrons unequally, so they're partially charged. So because of that, they're called polar molecules, and glucose will move through glucose transporters. Water also has specific transporters because water is partially charged. They're called aquaporins. So those are pores to transport water. Some channel proteins are open all the time, and the movement just depends on things moving from where there are more particles to less particles. Aquaporins are open all the time. There are certain ion channels that allow muscle contraction to happen. They're just open all the time. Some proteins are carrier proteins. That just means they alter their shape a little bit when things move through them. I would think about it being like somebody walking through a double door at the saloon. The double door at the saloon isn't being powered electrically. It's just your movement is kind of pushing through those double doors. Okay, so that's the story with a carrier protein. It doesn't use energy. There's no ATP involved. Here's a glucose transporter, but it changes shape slightly as the glucose molecule moves through it. There's a special form of diffusion for water. It's called osmosis. How does water move across a membrane? Water moves from an area where there are fewer particles dissolved in the solution to an area where there are more particles dissolved in the solution. So if this is a semi-permeable membrane, here you have fewer particles dissolved in the aqueous solution. Here you have more. So the water is going to move to the right. It's going to move until the particles are at equilibrium. Okay, so why do they say it's a specialized case of diffusion? We don't usually think of free water molecules, but some water molecules are bound to the solute and some water molecules are unbound. So the free water molecules are at a higher concentration on this side. The free water molecules are at a lower concentration on this side, so water moves in this direction. A lot of times people just memorize the definition as water moves from where there are fewer particles dissolved in solution to where there are more particles dissolved in solution. So whatever makes you remember it better, that's called osmosis. Okay, water movement's really important because cells are made mostly of water, and usually they talk about the solute concentration relative to the concentration of solute inside. Tonicity is the extracellular concentration relative to the concentration of particles inside the cell. So in the first situation, there are more particles outside the cell. It's a hypertonic situation, so water will leave the cell. In the second case, this is what animal cells like. Animal cells like to be bathed in a solution that's of equal concentration as what's in the cell. And so there's no net movement of water. Although water is moving in and out, it moves in and out equally. And finally, plants and fungi that have cell walls, like a hypotonic situation, where they're bathed in very dilute water. And the water moves into the cell and it pushes against the membrane and against the cell wall. So plants like to be watered with pure water. They don't like to be watered with salt water. So this is showing you the situation of organisms with cell walls. So here might be a plant or a fungi that has a cell wall, and they want to be watered with pure water, and water goes in. Animal cells like to be in an isotonic environment. Okay, they're just showing you, here's a plant that's wilted, and now they've re-watered it. If it doesn't get too wilted, you can re-water it and we'll come back. If it's too wilted, you're in trouble. This is a protease called Paramecium. Paramecium has more solutes in the surrounding freshwater solution. So water will move into the paramecium. And then there are special organelles called contractile vacuoles, which will pump out the extra water that's moved in through osmosis. Sometimes cells want to move things against a gradient. If you're pumping water uphill, if you're pumping something against a concentration gradient, if you're pumping something against charges where it would normally flow in a certain direction, you can do that in the cell, but it requires ATP. Sometimes ATP is used directly, and sometimes ATP is used indirectly. It's like your car is powered directly by gasoline, whereas water over a dam, it seems like, oh, it's just flowing freely, but maybe that water was pumped over the dam to begin with. So for proper functioning, animal cells maintain a negative charge inside themselves relative to a positive charge outside of themselves. And they do this by actively pumping in potassium ions and actively pumping out sodium ions. So we'll see in a minute how that works. But in any case, there's a pump, and this pump is an antiport, and it pumps out 3 sodium ions, and it pumps in 2 K positive ions. Later on, those ion charge differences can be released through other proteins, and when they're released, the positive moves towards a negative, and the negative moves towards a positive, and that electrical chemical gradient being released can do work. It's no different than a battery. So if one item moves in one direction, it's called a uniporter. If a molecular ion, if 2 are moved in the same direction, it's called a symporter. And when things are moved in opposite directions, it's called an antiporter. In the case of the sodium-potassium pump, that's an antiporter. So when things move in and out of cells, they move in and out based on a difference in concentration. We've seen that in terms of diffusion gradients, but they also move in and out based on a difference in charge. And when you're talking about the difference in concentration and charge together, we call that an electrogenic gradient. In this case, the way that cells maintain a negative charge inside is by this particular protein, which is a sodium-potassium transporter. And it shows the same protein in order. Kind of think of these as being sort of stop motion. So here are 3 sodium ions binding to the protein. Now ATP comes along, and ATP uses its energy to change the shape of the protein, and the 3 sodium ions are released outside of the cell. The 2 potassium ions bind, and then the ATP hydrolyzes, and the protein changes shape again, and then the 2 potassium ions are released inside the cell. So as this pump works, ATP is used to pump out 3 sodium and to pump in 2 potassium. So you're pumping out a charge of +3 and you're pumping in a charge of +2. And so because there are other ions on both sides, what's happening is you're pumping out positive charges and the outside becomes more positive, the inside becomes more negative. This is an antiporter. You're pumping out sodium and in potassium, and the pump is electrogenic. Electrogenic is changing both the concentration and the charge. So secondary active transport is just saying, well, gee, something's already been used using ATP to pump out all these sodium. And now as these sodium come in, they carry glucose along with it. So there's no ATP being shown directly, but the sodium was pumped out by ATP earlier. So it's a secondary source of transport. Okay, and it says that many amino acids and glucose enter the cell this way. The ATP creates a concentration gradient. In this case, it's a symporter that carries the glucose, these rectangular molecules, along with the sodium. All right, sometimes molecules are way too big to be able to move in and out through protein channels. So insulin, for example, is a protein. Proteins are made up of hundreds of amino acids, and a protein would not be able to move through a protein channel. So proteins need to move in and out through bulk transport, where the whole membrane invaginates through endocytosis, or it dumps a vesicle outside, exocytosis. Okay, so endo means in, exo means out. When we get to endocytosis, we'll divide it into phagocytosis, pinocytosis, and receptor-mediated endocytosis. All right, go ahead and watch this YouTube video. It's very cool, and it shows how a white blood cell, how the membrane surrounds a bacteria. That's phagocytosis, cellular eating, when the membrane surrounds a particle that it wants to get rid of or it wants to capture. Okay, pinocytosis. Pinocytosis is non-specific endocytosis. So in pinocytosis, the cell membrane invaginates, takes in fluid, and whatever happens to be there. And finally, receptor-mediated endocytosis, you're trying to uptake a certain molecule. So we'll look at all 3 forms of endocytosis. Okay, so phagocytosis, hopefully you'll click and see that little video from your downloaded PowerPoints. You're surrounding something, taking it in, and so it's cell eating. Pinocytosis, you're taking in fluid non-specifically, but you may be taking in particles as well, but you're just trying to bring some fluid in. And finally, an example of receptor-mediated endocytosis, sometimes cells need to bring in cholesterol. Cholesterol comes in bound to proteins. And so the cell doesn't want to invaginate until it has enough of those particles. So if the star represents a particle, if this blue Y represents a receptor, the cell might not invaginate until a certain number of receptors were filled, because it uses a lot of energy to bring in that particle to do endocytosis. All right, exocytosis is the opposite process. So let's say a cell in your pancreas is making insulin to be exported. That insulin is packaged in a vesicle. That vesicle merges with the plasma membrane. If 2 little oil droplets merge, you get a bigger oil droplet. So the phospholipid bilayer becomes part of the plasma membrane, and the particles are dumped outside. So that's exocytosis. Exo means out. Okay, that concludes Chapter 5. You should now be able to define the following terms, explain how membrane fluidity is influenced by temperature and membrane composition, distinguish between facilitated diffusion and active transport, explain how an electrogenic pump like the sodium-potassium pump creates voltage across a membrane, explain how large molecules like proteins move across a membrane, and here, tell me how would each of the following molecules cross the plasma membrane? Oxygen, glucose, insulin, and water. Okay, so oxygen can move by diffusion right across the phospholipid bilayer. Glucose has hydroxyl groups on it, so it's polar, partially charged, so it would have to go across a protein channel. Insulin is a protein made up of hundreds of amino acids, so it would have to leave the cell by bulk transport, by exocytosis. And water is partially charged, it's a polar molecule, and so it moves through a protein, and the name of the proteins water moves through are called aquaporins.